Hello brain nerds, my name is Jason. Welcome to the Big Brain Busters. So in this video we are going to look at the mechanisms of phagocytosis. Then as usual at the end of this video I've added some practice questions that I would like you to attempt and see whether you've understood. With that being said, let's jump into it. Talking of phagocytosis, this is simply a mechanism that is adopted by our body to help us fight against body infections such as viral, fungal, and bacterial. So we have three main steps of phagocytosis that are involved, and these steps are recognition and binding, engulfing, killing, and degradation. So the first one, which is recognition and binding, first of all, it makes sense because before you actually kill anything, you have to first recognize it. You have to see it to say, oh, okay, we have a microbe in the house. So now what's next? We go touch it, we go, hold it to prevent it from escaping. The next thing, we enclose it, we put it in a cell. That is where now engulfing comes in. Then now you sentence it to death, where now killing comes in. All right. Okay, so now let's look at the first step of phagocytosis, which is recognition and binding. Like, how do our leukocytes manage to carry out this function? Like, do they have certain types of eyes or special ears that they use to detect, to say, hey, look, we got a microbe in the house. Let's go exterminate it. So to explain this step, I'm going to use my diagram here, which shows a microbe and a leukocyte, which can be either a neutrophil or a macrophage. So now take note that in acute inflammation, the first cells in line towards the site of infection are simply the neutrophils, which are recruited first. Within 6 to 24 hours, carry out their function, but then they are short-lived, so they are degraded by apoptosis, and then they are replaced by what? Macrophages. Those macrophages have got a longer duration. Okay, so now let's look at what really makes them to carry out recognition and binding. So around the surface of the microbe, we have mannose and fucose sugar moieties. So these sugar moieties are the ones that make this guy to be super susceptible to phagocytosis, because that is what will make the receptors around the leukocytes to go and bind to these sugar moieties. Okay, then take note that these receptors that are found on the surface of the leukocytes are only able to recognize the dead necrotic tissue or the bacteria, virus, or fungi. And that is very important because you want to only remove the disease causing agents or the problem causing agents in your body. You don't want to start fighting against your own host cell or your own host cells in the body. Okay, so that is important, because if the opposite happens, whereby your immune system starts fighting against your own host cells, that's when now we have immune mediated infections or disorders. Okay, so now around the surface of the neutrophils, we have mannose receptors, scavenger receptors, and opsonine receptors. These mannose receptors are the ones that will bind to the mannose and sugar and fucose sugar moieties. Okay, then we have also scavenger receptors and opsonine receptors. Now take note of the opsonine receptors because they bind to certain types of proteins known as opsonins. What are these opsonins? So opsonins are protein structures actually, which go and coat themselves around the surface of the microbes. Why is that important? Because this makes this microbe very susceptible to what? To phagocytosis. Now let's look at different types of opsonins. We have immunoglobulin G. So IgG will go and coat itself around the surface of the microbe, and then it's like you going into the jungle and then you coat yourself with a whole bunch of honey around you, and then you encounter a bear. Bears love honey, so technically you'll be the meal for that day. So that's same as here. The IgG and the products of the complement system such as C3b and the mannose binding lectin. These are the opsonins which will go bind themselves to around the surface of the microbe to make it susceptible to what? Phagocytosis. And then these receptors will go and bind there. So this is all about recognition and binding. Now let's look at the second step of phagocytosis, which is engulfing. So how does engulfing take place? So now that you have seen and held the disease causing agent, okay, so we have bound to the disease causing agent, which is a microbe. Next thing, we need to enclose it. We need to put it in a cell so that it does not escape. How does that occur? So the thing is, these receptors on the surface of the leukocyte will be coated with adapting proteins. So these adapting proteins will be coated there, and the next thing is that the clathrin proteins, these blue structures, will go coat themselves around the adapting proteins. Why is that important? It is these proteins that will start allowing to pull they'll start pulling the what? This surface, the cell membrane surface, towards the cytosol. Okay, and then as they are pulling, you are actually mediating endocytosis so that you pull this microbe inside the cell. Okay, so as that is happening, more clathrin proteins will start coating around the adapting proteins, and then you form a coated vesicle. Okay, so this coated vesicle here will need to be separated from the surface of the cell membrane. And how will that be done? There is a GTPase enzyme known as dynamin, which will come and facilitate the separation of the coated vesicle from the surface of the cell membrane. And then when that separation takes place, we have this coated vesicle, and now it is called a phagosome. Okay, so this phagosome now will be transported into the cytosol, and then as it is being transported, the coated proteins, which are the adaptins and clathrins, will start separating from the surface, and then they'll be recycled to go carry out their functions. Then this coated phagosome, or I mean this phagosome here, since now it is no longer coated, this phagosome is now ready to fuse with a lysosome. Why is it important to fuse with a lysosome? Lysosomes are organelles that contain other, they contain enzymes that will be very important in killing and degrading of these bacteria. So this is all about engulfing, where we, endocytose that microbe, and then take it inside the cytosol, where it will fuse with the lysosome to form a phagolysosome. So a phagosome and a lysosome fuse together to form a what? A phagolysosome. So let's look at the last stage, which is killing and degradation. So this is just simply a summary of everything that we've talked about. Recognition and binding, then endocytosis, which is engulfing, take it inside, then cause fusion of the phagosome with the lysosome to form a phagolysosome. So this lysosome contains lysosomal enzymes that will be fused with this guy to form a phagolysosome. Now this process of forming a phagolysosome sometimes can make some of these enzymes to leak out of the phagolysosome. And when they leak out, those enzymes are very dangerous because they'll start destroying whatever they find. So it is very important that this lysosome and the phagosome only fuse, and then this whole process of killing and degradation takes place within one compartment. Okay, so for killing and degradation, we are interested in a very, very, very important system, which is where now we involve certain types of free radicals. So we involve certain types of free radicals and enzymes that will be responsible for killing and degradation, and that is when I will zoom into this picture and this picture. Okay, so now to carry out the process of killing and degradation, it involves three things: reactive oxygen species, reactive nitrogen species, which are together collectively known as free radicals, lysosomal enzymes, which contain primary granules such as myeloperoxidase. We'll talk about that. It also involves deficins, it involves acid hydrolases, neutral proteases. Then, as I've already mentioned, to say the reactive oxygen species and the reactive nitrogen species form the free radicals. Then the next thing we talk about is the secondary granules, which contain these enzymes, which are lysozymes, collagenase, gelatinases, lactoferrin, plasminogen activating factor, and histaminase, alkaline phosphatase. Okay, so we are going to focus mainly on these free radicals. Then lastly, we are going to talk about lysosomal enzymes. Okay, so now let's look at this step of killing and degradation by formation of reactive oxygen species. So these rows are formed in presence of an enzyme known as NADPH oxidase. So this oxidase enzyme is a 7 multi-component enzyme whose components are spread in the cytosol of the leukocyte, and it is only after formation of the phagocytic vacuole that the components of this enzyme assemble inside this vacuole and then start carrying out its function. So what will happen is that this enzyme, which is NADPH oxidase, also known as phagocytic oxidase, it will oxidize NADPH to NADP+, and in the process it will activate the membrane oxidase. This membrane oxidase will facilitate conversion of oxygen to superoxide. Then superoxide will be converted to hydrogen peroxide via a process of spontaneous dismutation. And this hydrogen peroxide is a very weak free radical, so meaning that it needs to be converted to a potent radical. So in this case, it will be converted to what? To hypochlorite. This hypochlorite is a very important free radical which is responsible for killing and degradation. How does this occur? So in the lysosomes of the leukocytes, they contain azurophilic granules which contain enzymes such as myeloperoxidase. So this myeloperoxidase, in presence of a halogen such as chloride, it will facilitate conversion of hydrogen peroxide to hypochlorite. Okay, then also this hydrogen peroxide can also be converted to another free radical, which is hydroxyl ion, which in the process of Fenton reaction. So this process can only occur in presence of what? Of ion. Okay, so this is the whole process of formation of reactive oxygen species, and these two free radicals are responsible for halogenation or lipid peroxidation of the microbial substances. So they will degrade and kill the microbe. Okay, so that's it about reactive oxygen species. Now let's look at the next free radical, which is reactive nitrogen species, and this is carried out or formed due to presence of nitric oxide. Nitric oxide is formed in presence of an enzyme known as nitric oxide synthase, which exists in three forms, and these are endothelial nitric oxide synthase, neuronal, and inducible nitric oxide synthase. The first two nitric oxide synthases are produced in low amounts, and they are responsible for maintaining vascular tone. Okay, because remember that nitric oxide is responsible for what? For vasodilation. So the first two will produce nitric oxide to produce what? To maintain the vascular tone. The one that we need in phagocytic reaction of killing and degradation is inducible nitric oxide synthase, which facilitates the conversion or formation of nitric oxide from arginine. And this nitric oxide will react with superoxide to form peroxynitrite. So peroxynitrite is what is the reactive nitrogen species that we need for lipid peroxidation of microbial substances. Okay, so that's it about formation of the reactive nitrogen species. Now let's look at the summary of this guy. So at first we talked of recognition and binding, where our leukocytes recognize the microbe and then they bind to the receptors of the microbe. Then next thing, we require that microbe to be put in a certain cell or prison, which is known as a phagocytic vacuole, and that is known as engulfing. Okay, then after that it fuses with a lysosome to form a phagolysosome. Then after that we require now killing and degradation, which we mentioned to say killing and degradation occurs either by formation of reactive oxygen species. So formation of these species here requires an enzyme known as NADPH oxidase, which facilitates the oxidation of NADPH, and in the process oxygen is reduced to superoxide, and then superoxide via spontaneous dismutation it is converted to hydrogen peroxide, which is a very less, or in short it is a weak free radical, so hence it must be converted to hypochlorite via an enzyme known as myeloperoxidase in presence of a halogen, which is chloride. The next thing, we have reactive nitrogen species, which require inducible nitric oxide synthase to catalyze nitric oxide formation, which reacts with actually here it should be superoxide, and then the next thing that will be formed is a peroxynitrite, which will be formed. Then another thing is simply the lysosomal enzymes. We have two types, which are primary azurophilic granules and secondary granules. Okay, so all these processes of killing and degradation will lead to one thing, which is halogenation and peroxidation. Okay, so that's it about phagocytosis. These are some of the practice questions that I would like you to attempt and see whether you've understood. The first question until to the fourth question, please try to attempt and see whether you understood. Okay, so this is where we end our video. Don't forget to like, share, and subscribe to our YouTube channel, and also place your comments in the comment box. With that being said, we end here. Shalom, shalom.